Right, it's been a bit since I recorded here, huh? No matter, I will simply... and... Perfect! Not going to bug me later on in the day whatsoever, alright? That is fine. Alright, now then. Hi, welcome back to IB Bio with Aiden. It's me, I'm Aiden. We are going to be covering 2 units today. Both of them suck. It is signaling within and between neurons. There is so much vocabulary, and there are so many concepts, and I've got a total of 3 days before the test to pump this one out, so let's just get into it. I think the best way to start off is, what is a neuron? Now, to improve upon my last video, which I will admit was kind of garbage, I am going to be enlisting the help of some diagrams, like this one. Yeah, isn't that cool? I got pictures now. Please do not say it's uncool. This means a lot to me, okay? Anyways, this is a neuron. Now, hard as this may be to believe, my wall here is not a neuron, so I can't physically point to any part of what I'm talking about of the neuron. So instead, I will simply circle in red whatever I'm talking about. If that doesn't work for you, I'm sorry. I really don't feel like coloring my whole wall into a neuron. Sounds like a lot of work. Now, what we're going to do is first off go over the stuff that doesn't really do a whole lot for signal transmission. So we're going to start off with the cell body and nucleus. The cell body, also known as the soma, while important for keeping up the life of a neuron, is not very important in signal transmission. All it does is it lets the signal go past, which kind of describes the entire neuron, so it's not worth saying a lot. And the nucleus in a neuron does the same stuff that a nucleus does in pretty much every cell. Directs and overall controls the cell as a whole. And with that, we are also kind of out of the parts of the cell that don't really do a lot for signal transmission. So we'll go on to the dendrites, since they tend to be at the top of every diagram, and we will work our way down. So the dendrites, they tend to be the first signal reception site. So when a signal or neurotransmitters comes from another cell, they tend to flow into the dendrites, where those signals and or neurotransmitters then get sent down to the axon hillock, which we will go over later. It's important for another step of the whole signal process, which we'll just cover that later. Moving on from the axon hillock, we've got, and you can probably guess this for yourself, we've got the axon. Now, the axon itself doesn't really do a whole lot. It's what's on it that is important. Pretty much the only thing that the axon really does is it kind of extends the signal through to the axon terminals, which we will go over very momentarily. In sensory neurons, you will tend to see that they kind of split off into 2, kind of like this. But what's important about the axon is not necessarily what it does, it is what's on it, which are myelin sheaths. Myelin sheaths are made by Schwann cells. What are Schwann cells? Well, they are glial cells, which means this, that form around the axon, and they will produce layers of lipid, which eventually, with enough layers of these lipids, form the myelin sheaths. Now, these sheaths, they act as an insulator, which, if you know anything about electric signals, seems pretty counterintuitive to throw in a bunch of insulators when you're trying to transmit an electrical signal. Well, it somehow actually boosts the process through saltatory conduction. What is saltatory conduction? Now, saltatory conduction is basically the process of an electric signal made by a neuron passing through each of these myelin sheaths into these nodes of Ranvier. Don't ask me about the name. Scientists are weird and they name things weirdly, okay? This is tip of the iceberg, really. This whole process results in a much faster signal transmission than just sending it on down the axon like you would expect. How, you may be asking? How does this happen? Well, all you gotta know for right now, to keep it as simple as possible, I'm gonna get a little unscientific in how I describe this, but just stick with me. So basically, each of these nodes of Ranvier here, since they aren't insulated, they've got a lot of electric potential. So when an electric signal is, coming on down, trying to go to the axon terminals, it sees each of these electric potentials and it hops to there instead of trying to go through the insulated space. And since that potential is gathered at each of these spots, it effectively fuels this hopping, and the hopping is a lot faster. How much of a difference is there between these saltatory conductions and regular non-saltatory? A lot, actually. It's kind of impressive. So myelin sheaths are highly important to keeping the nervous system and just nerves as a whole moving quickly, which is very important for, doing anything. Okay, and we have reached the end of the neuron. We are at the axon terminals. Now the axon terminals, these are where the whole electric signal will end up at, and it will open up calcium channels, which will cause neurotransmitters to leave and go on to the next cell. Now, whichever cell now that we know what a neuron is and all of its parts, we can move into its signaling. So the way that a neuron sends signals is it generates an action potential. Now, these action potentials are electrical signals that will be sent from the dendrite all the way down to the axon terminal, and then from there it will, activate neurotransmitters. They go into a new cell. Those do their own thing. So, how do we make an action potential? Well, before we can actually go over how to make an action potential, we have to go over what a neuron is like at a resting state. Now, at a resting state, a neuron is polarized. That just means that there is an imbalance between the positive and negative ions that are either inside of or outside of a cell. For the case of neurons, they are almost always negatively charged. Now, granted, they are positively charged during this brief window of making an action potential, but this is also just when the action potential is made, so I don't know how much you really want to count that. But for the most part, they are almost always negatively charged, and at a resting state, they are at negative 70 microvolts. Now, how are they always this negative pretty much the entire time? Well, it is through sodium and potassium pumps. The way that these sodium and potassium pumps work is that for every 2 potassium that brings in, it will pump out 3 sodium. Now, both of these ions are positive, but since we are pushing out 3 sodium for 2 potassium, the outside of the neuron is going to be relatively more positive than the inside of the neuron, making the inside of the neuron relatively negative. With this, there's also the potential for some potassium ions to just sort of leak out of the neuron, making it more negative because there are even less positive ions inside the neuron. And there are also some negative proteins that are inside the neuron, making it even more negative. Now, I don't know how important this is going to be in the realm of IB biology, but if you've been keeping track, you've probably noticed that the pumps kind of work a lot because you have to keep that negative potential pretty much the entire existence of a neuron, which means that these pumps are going all the time. And these pumps, they use ATP to work. So if you've ever heard the fact, brains require a lot of energy to work, this is kind of the reason. They require ATP to work pretty much, because a neuron isn't going to generate an action potential if it isn't negative to start out with. And that negativity is only really maintained through these pumps. So without all this energy going through to the pumps, your neurons don't do anything, and your brain is filled with neurons. That's why a big brain kind of requires a lot of energy. There are so many neurons that require so much ATP, which is so much energy, that in the realm of biology, it's a lot of energy to have a bigger brain. I don't know if that's going to be important for anything, but, keep it in mind. I don't know. You do you. In addition to these pumps, we've also got gated ion channels, which also help move potassium and sodium either inside or outside of the neuron. What does gated mean? Well, it just means that the channel can either be opened or closed depending on some sort of factor. In the case of neurons, it is the voltage of the neuron that will dictate whether or not these channels are open or closed. Alright, so we've already got all these pumps that are moving potassium and sodium in and out of the cell. What do we need these channels for? Well, the channels are actually very vital for creating the action potential itself. It is what gets our neuron to being at a positive voltage. Now, remember how we've been moving a lot of sodium outside of the neuron? Yeah, well, once we use these channels, they are passive transport, meaning that we go from the high concentration of sodium outside of the neuron to the low concentration of sodium within the neuron. Basically, what I'm saying is that once these channels open, we get a bunch of positive ions that just fly right into the neuron. This will create the action potential. This will make our neuron way more positive than it was before. So how do we open it? Well, we've just got to get our neuron to about negative 55 microvolts, because that's at the point where it will open up. So how do we prevent it from just forever being opened up at this point? Well, they will close at around 35 microvolts. Just check my notes. 30 microvolts, not 35. Moving on. We don't just have sodium channels though, we've also got potassium channels. Now, these ones will actually push potassium out of the cell rather than bringing them in. Why? Well, it's because we've been bringing in all of this potassium already. And this whole process is done after the action potential is made to get our neuron back to its resting potential of negative 70 microvolts. We need to bring it back down, so the best way to do that is to get all of these positive ions out, such as our potassium. We will also get rid of all of our sodium, but that's through the pumps. The channels, however, are what will get our potassium out to quickly bring us back down to these negative 70 microvolts. So if you've been keeping track at home, you might have noticed that we start off at negative 70 microvolts, and we need to get to negative 55 for the neuron to do anything. So how do we get that increase of 15 microvolts? Well, it is going to be via one of two things. For right now, it's going to be our stimuli. Later on, it's going to be neurotransmitters. We'll cover that in however many minutes we do. So for the stimuli, it's pretty much any sort of change in the environment that will cause an electrical impulse. To keep it simple, basically it is anything that your sensory neurons will pick up on. It's going to be stuff like mechanical things, like being pinched or the pressure. It is going to be thermal stimuli, such as intense heat or cold. And then there are also chemical stimuli, such as acids, alkalis, irritants in general, that sort of stuff. These stimuli will help generate a bit of a response that will cause us to get to that threshold potential of negative 55 millivolts. Microvolts. Negative 55 microvolts. And now, it is the part that I've kind of been dreading doing. It's walking us through an entire chart of this entire process. Alright, so here we start off at our resting potential. We start off at negative 70 microvolts. Nothing's happened yet. Our neuron is just living its best life, pumping out sodium while it takes in potassium. At this point, we get our stimulus. This is going to be that either mechanical or thermal or chemical stimuli that causes a reaction. So basically what the stimuli does is it is going to cause some sodium channels to open up, causing a bit of positive ions to enter the neuron, which will increase our voltage, and hopefully it will increase it to the negative 55 microvolts, which is our threshold potential. Once we have hit this point, all of those voltage-gated ion channels for the sodium are going to open. That means we get a bunch of positive sodium ions to enter the neuron, which is going to cause this section called the action potential. This is where the neuron sends out that signal. It is our point in which we are fully positive, and this whole section is called depolarization because we no longer have an imbalance in positive and negative ions right here, and then we just become positive here. That's the best way I can describe thinking about it because it is a bit of a weird term. We go positive during depolarization. That's what you've got to know. Now, we can't do this forever. It's not exactly great to just constantly send out action potentials, and it also isn't all that manageable. So what will happen is the repolarization stage, in which all of our potassium channels are going to open. And we are going to take all of these positive potassium ions outside of the neuron, which will bring us back down even a bit lower than our negative 70 microvolts. Us being below the negative 70 is called the hyperpolarization stage, and all of this kind of makes up the refractory period in which we go below our negative 70 and we come back up to it. And then at that point, we reactivate our pumps and keep us at a negative 70 microvolts level until the next stimuli comes along, and then we repeat the whole process again. Thankfully, we're through with the bulk of all of the signaling within neuron stuff. We've got a few more things to cover, but they're pretty light, such as how do we get these lovely graphs of our action potentials? Well, it is through oscilloscopes. Now, what these do is they pretty much just record the electrical voltage of a neuron over the amount of time. That's how we get these sorts of graphs here. There are a few factors that can affect the speed at which our nerves send impulses, one of those being body size. Now, what happens here is that there will be a bit of a delay in signal transmission the larger a creature is. Now, if we look at an example for this, we can see that the main reason is just there's more ground to cover. If we look at an elephant, for example, if we are looking for a signal to be transmitted from its head to its foot, we've got a lot more distance that we gotta cover than that in a mouse. Another thing that can affect signal transmission is axon diameter. For the most part, a larger axon diameter will result in faster signal transmissions because there's less resistance to the ion flow. And the last main factor that will affect our nerve impulses is myelination. This is how many myelin sheaths there are, how much our action potential can jump between these myelin sheaths. Pretty much the more myelin sheaths there are, the faster that it can do this because it can just jump between all these nodes of Ranvier way faster. Speaking of myelin sheaths, this is where we get a good example of a disease that really affects our signal transmission. Multiple sclerosis will attack our Schwann cells. Schwann cells, if you remember, make our myelin sheaths. Myelin sheaths are important for making saltatory conduction possible. Now, some of you might be thinking, well, it's fine if we don't have these Schwann cells to make myelin sheaths to do saltatory conduction because we can still send a signal without those. Certainly it's fine, right? It's still survivable. Not really. The thing is that saltatory conduction is about 100 times faster than its non-saltatory counterpart. So without these Schwann cells and saltatory conduction, it's pretty much an unsurvivable situation if too many of these Schwann cells are affected. That's why multiple sclerosis is a very deadly and very dangerous disease to develop. And that is everything for signaling within neurons. We've got signaling between neurons, but thankfully it isn't too many new things. It's mostly just throwing in neurotransmitters into what we have already learned. So, let's get into it. Now, a huge part to signaling between neurons is the forming of synapses. Synapses are effectively just the connecting point between a neuron and another cell. Now, the space in between this neuron and another cell is referred to as the synaptic gap or a synaptic cleft. Most of the time, synapses are formed between a neuron and another neuron. You will see the axon terminal go to the dendrite of a different neuron. However, this is not always the case. You can see other forms of these synapses. One good example is sensory cells, such as photoreceptors in your eyes or hair cells in your ears, will connect to neurons that will then transmit the information to your brain. This is how you get your sensory organs to transmit any sort of info to your brain to actually process it. Another thing that you can see is the connection between a motor neuron and a muscle or gland. This is how you get your muscles to do anything or a gland to work. It's important to note that for people, our signal transmissions usually only travel in one direction, from the dendrites to the axon terminals. This is because our axon terminals are where neurotransmitters are sent out from to a new cell. In this case, for the neuron, the new cell is going to have the dendrites pick up these neurotransmitters because they have receptors for the neurotransmitters. It wouldn't make any sense for us to send a signal from the axon terminal to the dendrite because there's nothing to send out from the dendrite. You follow me? This is also why the axon terminal is our presynaptic cell. It is part of the cell before anything with the synapse actually happens, before we send out any neurotransmitters. This is why our dendrites are the postsynaptic cell. They take in the neurotransmitters that travel across the synapse, so they come after everything involving the synapse. Now, I've been talking about neurotransmitters a bit, but what are they? Well, neurotransmitters are just chemical messengers that have very localized effects. They are sent across the synapse via diffusion. That is because their high concentration is at the axon terminal, and they will go to a lower concentration, which is the new cell, specifically the receptors for those neurotransmitters. Unfortunately, we do have to go over how we move neurotransmitters outside of the cell into a new one. So let's just start out. So first off, this is where the knowledge of signaling within a neuron is actually kind of important to know. We start off with a nerve impulse being sent all the way through to the axon terminal. This happens in the presynaptic cell. From here, the depolarization that we have from generating this nerve impulse will cause voltage-gated calcium channels to open. Note that I said calcium, not sodium or potassium. Those are just for generating the action potential. Calcium is what we're going to be using to send out these neurotransmitters. Now, this influx of calcium ions into the neuron is going to cause vesicles containing the neurotransmitters we want to go to a new cell to go to the axon terminal of our presynaptic cell. From here, they will then leave the presynaptic cell via exocytosis, and they will travel through the synapse into the receptors of the next cell, our postsynaptic cell. Now, there are usually just two types of neurotransmitters, excitatory or inhibitory. Excitatory means that it will cause something to happen, meanwhile inhibitory means that it will prevent something from happening. To give some examples of each, acetylcholine is excitatory. It is responsible for muscle contraction, among other things. And GABA is inhibitory. It will depress the activity of your postsynaptic cells, the ones that receive it. Now, we're going to go over which major types of neurotransmitters are inhibitory and excitatory. So the big four that you're going to see a lot is GABA, serotonin, dopamine, and acetylcholine. These are the four main neurotransmitters that you'll see the most commonly. So GABA is inhibitory. We already covered that. Dopamine can be either excitatory or inhibitory depending on where it binds to. Serotonin is inhibitory and acetylcholine is excitatory. Alright, so now that we've gone over our excitatory and inhibitory neurotransmitters, how do they actually do anything? What actually makes them cause any sort of difference in the postsynaptic neuron? Well, it's because of more gated ion channels. In addition to the voltage-gated ion channels we discussed previously, there are also ligand-gated ones. This just means that they will open in response to a certain chemical compound or element being in presence with the receptor. In this case, it will be our neurotransmitters that we're sending out. These neurotransmitters, usually just the excitatory ones, will bind to these receptors, cause them to open, and then sodium or potassium will enter the neuron, which will then increase our voltage. This is why they are excitatory. It will cause more action to possibly happen from our neuron. It will increase the voltage, increase the likelihood of us generating an action potential before we go back down to below our threshold potential. Our inhibitory neurotransmitters too can interact with these ligand-gated ion channels. One of the two main ways that this will happen is they will interact with a potassium channel, which will then let out potassium during the refractory period because the inside of our neuron has more potassium inside of it than outside, so by opening this channel, a bunch of potassium will leave, making the inside of the neuron more negative. Another thing that you will see is instead of a potassium or sodium channel being affected, it will be a chlorine channel. Why chlorine? Well, because it's negative. So this channel will open up and chlorine will go into the neuron and make it more negative. This is how these inhibitory neurotransmitters can inhibit the generation of an action potential in another neuron. And now it's time to put all the pieces of the puzzle together. Each of our neurotransmitters effects on where sodium and potassium and chlorine goes in or out of the cell. It is the process of summation. Effectively, just how each individual neurotransmitter has its own effect on the overall voltage of the neuron. Also, by the way, you remember when I said that we were going to talk about the axon hillock later? This is when we talk about it. The axon hillock is where all of summation happens. That's pretty much it. The important thing about summation is that it is the effect of all of our excitatory neurotransmitters and the effect of all of our inhibitory neurotransmitters combining together to see if there are more excitatory neurotransmitters that will cause an action potential in the neuron. Because if there aren't enough of these excitatory ones, then the inhibitory neurotransmitters are just going to lower our voltage and we'll never reach the threshold potential and thus never make an action potential. That's what this has all been leading to. All of these neurotransmitters basically dictate whether or not we are going to see an action potential made by all of these neurotransmitters. We have to talk about acetylcholine now. I don't want to, but I have to. So, acetylcholine comes from choline and acetic acid. Choline will come from your diet, and acetic acid is made when an acetyl group, which comes from cellular respiration, interacts with acetyltransferase, and thus you get your acetic acid. Now, these two form together, and you get acetylcholine. When we use acetylcholine as a neurotransmitter, it will bind to a ligand-gated ion channel. Remember that these ion channels are just ones that will open up in response to a neurotransmitter, and then sodium will enter through this channel. If there's enough sodium and potassium and all that good jazz, then an action potential will be fired off. Now, after acetylcholine has done its job, acetylcholine esterase, which is an enzyme that breaks down acetylcholine, will break it down into choline, which just gets recycled by the presynaptic cell, and then acetate. Acetate will just be excreted as waste because it's not really useful anymore. So why do we have to get rid of these acetylcholines on the receptors? Why do we have to break them down? Well, it's because if they remain there, they will be on the receptors indefinitely and cause several action potentials to happen, which isn't necessarily a good thing. That's why we have the entire repolarization stage. It's bad to just constantly have action potentials firing off. So by breaking it down into choline and acetate, they can't activate these receptors anymore, and now we don't have to worry about them causing an indefinite amount of action potentials. Acetylcholine is one of the most seen neurotransmitters in biology. It is highly seen in invertebrates and just vertebrates. It also does more than just the muscle contraction that we discussed earlier. It is also important for your memory, for your motivation, your attention, your learning, your arousal, and REM sleep. So now that we went over acetylcholine and what it does, all that, we can now move on to an inhibitory neurotransmitter and look at it more closely. For this, we're going to be looking at GABA, also known as gamma-aminobutyric acid. I may have butchered that, and if I did, you guys got the Google version of it. Now, GABA is one of the most common neurotransmitters in the brain. It is highly important for keeping you from effectively going insane by having too many action potentials happen. One of the main things that GABA is responsible for is reducing the ability for a neuron to receive, create, or send new signals. This, in turn, can reduce stress, relieve anxiety, or result in improved sleep. And now we get to talk about alcohol. Now, alcohol can cause more GABA to be present in your brain, which, not problematic in, normal amounts. It is very dangerous once you get to the alcohol poisoning levels because there is so much GABA present in the brain and kind of stops your neurons from actually doing anything signal-wise. That means you effectively go brain dead if you've got too much GABA in there, and going brain dead will just make you dead. So, drink responsibly or don't drink at all if you're below the age of 21. You wouldn't do that, right? You're all responsible adults, right? Right. Now I'll get off my soapbox about alcohol and all that, and we're going to move on to exogenous chemicals. Now, this just means any sort of chemical that doesn't come from within the body, and there is a large list of them that are possible. Like these ones, because I don't feel like going through all of them. They're pretty straightforward. It's just anything that can introduce you to new chemicals or compounds that you aren't normally exposed to. Now, pretty much these exogenous chemicals can affect every step of the neurotransmitter production process and sending out process. And they will either cause them to be excited or inhibited. They will either increase the amount of neurotransmitters you make or store or send out or this, that, or the other, or they will do the opposite. It pretty much depends on what you've got. Now we're going to move into two main examples. Now, one big example of inhibitory exogenous chemicals are neonicotinoids. Now, these are commonly used as pesticides, which what they do is they will block the reception of acetylcholine. Now, we went over how acetylcholine is very vital to, your function, how it's one of the most common neurotransmitters used pretty much across the board, right? Well, without all of this, muscle contraction isn't really possible along with all the other stuff, which results in paralysis and death in insects. Which does not sound fun. Thankfully, it's just used as an insecticide, and I think that it is safe for humans to be around. I think. Now, for an excitatory example, we've got, I don't know if I can say it, so, nose powder. Nose powder, you guys picking up what I'm putting down? Scarface. Scarface nose powder. I hope you guys understand what I'm trying to say here, okay? Now, what this does is it will block the reuptake of dopamine. Reuptake is just the taking back of a neurotransmitter into the presynaptic cell to prevent it from constantly having an effect. That's kind of what we did with acetylcholine after it did its job. We used the acetylcholine esterase as a way to reuptake acetylcholine and get it out of these receptors. Well, by blocking the reuptake of dopamine, that means that dopamine just remains in these receptors indefinitely and keep having their effect. And since they are excitatory, that means we keep having them do excitatory stuff. That's kind of why people get addicted to it. It's kind of why it's a very prominent illegal substance. Okay, so that should be about everything, or at least everything important. I left out a few things because I felt as though they were either redundant or just not that important. So, hopefully this will help you if you have a test coming in the near future, which I know I do, hence me making this video. And unless I've left something out and am about to talk about it right now, this should be the end of the video. So, thank you for watching. Subscribe or don't. Actually, never mind. Subscribe, like, comment, favorite this video. Is that even a feature? Do everything. Alright? I want 1 million views by the end of tonight, okay? Anyways, thank you for watching. This has been IB Bio with Aiden. I'm Aiden. I was Aiden. I will see you next time, which could either be a few weeks from now or a few years. See ya!